Hello everyone and welcome back to the lovely bio YouTube lecture series. I am still Serene and I got new nails. Look, they're plant themed because today we're talking about photosynthesis, part 1. Today we are talking about the light reactions of photosynthesis. Today I will be using 3 colors, so you can do that too, and a pen, and possibly whiteout if we make mistakes. Let's hope not. All right, let's get into it. Okay, photosynthesis. So, basically, you've probably got the bare basics of plant, turn light into food, somehow. So we can actually break this down into an equation. What exactly is happening here? So what exactly a plant does, or not just plant. Actually, let's talk about who does photosynthesis. It's only done by autotrophs, or like heteroautotrophs, which are like a type of autotroph, like they're both auto and heterotroph. But we went over that previously. If you didn't catch a previous video about bacteria, you can check that out. But it's only done by autotrophs. So those are the producers, right? And here, what we're producing is glucose. We're making glucose. Slay. Okay. And so you're like, well, yeah, plants, right? But you'll also see things like cyanobacteria do this type of bacteria that photosynthesize. And they're called the blue-green algae, but cyanobacteria are not actually algae. They're just called that. But they're cyano, or blue-green, because that's the color they are. They're like a teal, but usually more green. And also algae. So there are protists. Protists are a type of single-celled organism, and the ones that photosynthesize are called algae. And that's what algae is. Fun fact, not a plant, technically. Okay, so what is this equation? How are we chemically converting light into food? So we start with some light, and we also have some CO2, carbon dioxide, and this is in a gas form. So this little G represents gas. So the cool thing about plants is they have these things called stomata, which are basically like little pores in on the surface of a plant cell, or a plant, not, it's made of cells. And so you can have stuff like carbon dioxide go in through the stomata, or technically this is just one, so it's a stoma. If you have multiple, they are stomata. So this like pore is open, and it can close up when it's really hot, because it can also emit water and oxygen. If things are really hot and it's releasing a lot of water, then the plant can like have problems. You know, it's going to shrivel up. So it can sometimes close the stomata. But right now, the stoma is open and it's absorbing some carbon dioxide gas. Because carbon dioxide is like what we exhale. Living organisms metabolize their food and as a result produce carbon dioxide. It's going through the stoma. So now we have some carbon dioxide, and then we also have 12H2O. Hope you know what H2O is. This is in the liquid form. Some water. So this comes from the roots of the plant. You know, the roots absorb water. They do that in plants. And it goes to leaves via transpiration. And you say, what the frig is transpiration? I'll tell you. Okay. So basically, you have your plant, and there is water in the roots, and then there's water in the leaves too, and the water evaporates, usually through the stomata. And so like, imagine it could also be going this way. And when that happens, it like pulls water like in the roots up to the leaves. This is called capillary action. So water evaporates, and more water travels up to leaves via capillary action. So kind of think of capillary action as like a straw, like, you know how you'll like suck on a straw? Like if you drink the water at the top of the straw, then like the rest of the water will come up. Basically, the evaporation of the water through the stomata is sucking up the water at the roots, and then we get it at the leaves again. And so there's water in the leaves now. And then if we add some enzymes, we can have photosynthesis occurring. So these are the reactants, the things that react to make the products. And so our products are C6H12O6, also known as glucose. And you usually see this represented like this. I don't know if you need to know that, but that's what it looks like, the simplified version. And then C6H2O, and it'll be emitted as a gas. And 6O2, also as a gas, and this will exit. And you'll also get some heat in there. Okay, so like I said, these are the reactants, and they react to form the products. Cool. So this is our overall reaction. But it's not that simple. You can't just say, okay, do that. You know, I'll give you some light and CO2 and H2O, and you can do that. It has to go through a couple of steps. Okay, before we look at the actual reaction that's happening, let's take a look at the chloroplast. So, what is a chloroplast? What does that look like? Guys, ignore my little whiteout moment there. Okay. So, the chloroplast has two layers. If you remember our lovely endosymbiotic theory, we think now, currently, that chloroplasts evolved from a cyanobacterium, actually, a cell enveloped a cyanobacterium that was, doing photosynthesis. This is like our chloroplast in a plant cell. And when that happened, another layer of membrane formed. So this is the chloroplast. Chloroplast. So now, not only do cyanobacteria have chloroplasts, we have them too, says the plants. Oh, that was totally intentional. Okay, so what else do we got in here? Okay, well, there are these stacks of I don't even know what you would call them. They look like pancakes. Sometimes they're drawn where they're like folded over like this. Some of them. Sometimes they're folded over like, okay, are you ready for this? It's folded over like 3 times. But you can just draw them as circles, it's fine. So we're gonna get comfortable with all the stuff. And sometimes they're connected. There will be these here, I'm gonna connect this one to that guy. Okay, so what is happening? What are these discs? So a single disc is called a thylakoid. This is a single disc, and a whole stack of them is called a granum. And if you have several stacks, the plural of granum is grana. So then we have 4 grana in this chloroplast, which is not very many. This is not to scale. Usually you would have more. And then these little connecting lines are called lamella. And then there's like a fluid space inside of the thylakoid, and that's called the lumen. Inside there. And then there's also a fluid outside of the thylakoid, but inside the membranes, and this is called the stroma. Okay, this is the inner membrane, and this is the outer membrane, which I know, that was really hard. And if you also want to label it, the space in between is the intermembrane space, but I think you got it. Okay, a little review. This is a granum. It's a stack of thylakoids, and then a thylakoid is this guy. The space inside of the thylakoid is the lumen, and the space inside this, the chloroplast, the whole chloroplast, but outside of the thylakoid, that's the stroma. The little intersections between grana are called lamella. They're connected. They can transport stuff to one another. The inner membrane is the inner membrane, and the outer membrane, That's the outer membrane. I know, that's really cool. Okay, so now that we have the basic structure of the chloroplast down, let's take a look on how it actually does photosynthesis. Okay, I just filmed a bunch and you couldn't see it all. So you could barely see any of it. So photosynthesis continued. So we looked at our chloroplast. Now let's see what's happening in the thylakoid membrane to be, you know, making food from light. Okay, so first we're gonna draw the sun, and it's, you know, emitting little photons. Photons are like the packets of light, like the energy. It was widely debated. Is it a particle? Is it a wave? Turns out it's both. So it's emitting photons, very exciting. And now I'm gonna draw some shapes. So we're looking, like, imagine we're looking right here, we're zooming into like this region of the thylakoid. And there's like different components of the thylakoid membrane. And so I'm just gonna draw the basic shapes. And you can too, but you're not gonna understand what any of them are until I explain them. This is supposed to look kind of like a hot dog bun. Okay, it's not supposed to. I just think it looks like a hot dog bun. And this looks, I don't know, like a really skinny football. We don't need to be describing it. This is just the shapes based off of images I've seen in a textbook, because believe it or not, I don't know exactly what these complexes look like. I mean, we don't overall really know what these complexes look like. It's all kind of like computer generated stuff because it's just so small. How do we see it? But I mean, we know it looks like this, but like, I can't show you like a picture just really zoomed in, you know? Okay, so we have 2 hot dog buns and a really skinny football and like a ring of squashed little other rings, and like a tube kind of is the situation. And you're like, what is going on? Totally valid. Totally understand the questions you're asking. But first, let's just draw our phospholipid bilayer, because you know we're having the thylakoid membrane. So membrane made of phospholipids with our polar head and non-polar tails. Do we remember that? We remember that? And it does not have to be super duper, I don't know, neat, complex, accurate. It just kind of, we gotta know, like, it's a membrane. So it's separating our lumen in here, inside, to the stroma outside in the chloroplast. So that's what this membrane is doing. Okay, so what are all these pieces? We'll draw like one more here. And so it's like curving. So it's like we're like right here, you know? Oh, I just drew on the paper. Not recommended. You do not need to do that. Okay, so this hot dog is something we like to call photosystem 2. And you're saying, photosystem 2? Where's photosystem 1? Well, it's over here. This is photosystem 1. And you're saying, what? Why is photosystem 2 before photosystem 1? Well, basically, we found this one first. We found photosystem 1, and we named it, and we're like, wow, this is awesome. And then we found photosystem 2. But we already knew this one first, so even though this one happens first in our little process of photosynthesis, it is photosystem 2. So that's annoying, but you gotta live with it because that's what it is. And also sometimes you'll see it notated like this, PS2. This is like a Roman numerals for some reason. Why? No say, but that's what it is. Okay, so our photosystem is absorb this light energy so we can make eventually our glucose, but it's a long process, so don't get too excited. Okay, so get very excited. It's very exciting. So the photosystem has a bunch of pigment molecules on it, and these are what's absorbing that light energy. So these are pigment molecules. So an example of that is chlorophyll. Have you heard of chlorophyll before? Very popular type of pigment molecule. So example, chlorophyll. Awesome. Oh, and if you wanted to label it, which I highly recommend, this is the outside of our thylakoid. This is the stroma, the fluid inside the chloroplast. And this is the inside. This is the inside fluid called the lumen. So let's just label that so we know where we are. Okay, that beside the point. So, okay, there's a lovely ray of light. So we're absorbing a photon, or maybe several. When we absorb a photon, there's a lot of energy that's being absorbed, and that can excite the electrons in the pigment molecules, and it'll travel this energy, boosting energy levels from pigment molecule to pigment molecule, and eventually what will happen is it'll go over here. Oh, this does not look very circular. To these two molecules are two chlorophyll A molecules. This is specifically chlorophyll A. So we're over here, it could be other types of pigments like carotenoids or anthocyanins. This is specifically chlorophyll A. Okay, so what's gonna happen is this high energy is gonna transfer over here, and it's gonna be absorbed by one of these chlorophyll A molecules, and we're going to take electrons from something, mystery what's it gonna be? And these electrons are gonna join the chlorophyll A molecule. So when we transfer over this energy, it'll boost these electrons that we're getting to a higher energy level. So where are the electrons coming from? It's coming from water. And then if you split, like, you know, like the structure of water, H2O, where you have the hydrogen bonding to the oxygen. Well, imagine you're taking the electrons away from that bond. So then you get 1/2 O2 plus 2H plus. And I know that the 1/2 O2 sounds counterintuitive, but remember, we're emitting this as a gas, so the O2 is the form of the oxygen. So we're gonna be making oxygen gas. So our H plus is a proton, and that will be joining the lumen where there's actually a lot of protons going on. We can draw more of these later, but it's very acidic in here. It's acidic. We'll draw more of these later. But we strip away the electrons, and that's gonna go up and be accepted by what? An electron acceptor. Wow. Creative name, guys. That's okay. Less things to be confusing to memorize. So I'm gonna put this arrow over here. This is an electron acceptor. And real quick, if you're in AP Bio, you probably don't have to know these terms. But if you're in botany or Gen Bio, you wanna know that this reaction where we're splitting water and we're reducing this electron acceptor, this area where this all is happening is the reaction center. Yay, a name that makes sense. And then, okay, this one is a little less intuitive. This area here where we have the pigment molecules, like here, it's technically this side too, we're just only drawing it on this side. This is the antenna complex. Okay, so in a photosystem, this is photosystem 2, for example, but this also applies to photosystem 1, we just haven't got there yet. We have the antenna complex filled with pigment molecules that's collecting solar energy, so you can say collects solar energy. And we use that high energy to boost the electrons that we get from water into a higher energy level. And then what we do is we move them over here. And you're like, what is that? It's a circle. It's my best friend PQ. And you're like, what is PQ? That is so valid. It's a great question. This is plastoquinone. And the names of these things probably don't have to know for AP Bio. In fact, definitely don't have to know for AP Bio, but it is really helpful if you're going to take a college level class. And there's a lot of other stuff here that's really important. So even if you are just in AP Bio, would recommend to know these terms. So the electrons will go through plastoquinone. You don't really have to know what it does. It just does. Okay. There's a lot of things where it's like, it just does that. Okay. And it'll go through the cytochrome B6/F complex. That's this thing. We'll label it over here. Not right here. Don't label it over here, and I'll show you why. So this is the cytochrome B6/F complex. And this thing is a proton pump. So you know how we have a lot of protons here, hydrogen ion where it has no electrons is a proton? Because there's no neutron in a normal hydrogen isotope, so it's just a proton when there's no electron. So there's a lot of protons in here, and that's really good. Like, we want it to be like that, and I'll explain why soon. But basically, so these super high energy electrons are going to be used by the cytochrome B6/F complex to pump a proton in. So there's a couple protons in the stroma, like a couple, like I'm going to draw 2. There's definitely more than 2, but like we want to make it look like there's a ton in here and like not that many in here. So the proton will go through here, and the cytochrome B6/F complex is going to use energy to push it through. So that's what we were drawing here. And why does it require energy to push this proton through? Well, there's a concentration gradient. There's a lot more protons in here and a lot fewer in there. So naturally, the protons are going to want to actually go this way, right? You think if you have a bucket of water and you put a drop of dye in, it's going to spread out, right? It's not going to stay concentrated in its tiny little droplet. It's going to want to spread. So that's kind of how the natural order of things works. That's our entropy. Might have heard about this before. And so it's going to really want to even out. That's just the natural order of the universe. And so it's going to require energy to force a proton in instead of not doing anything at all or letting it out. But letting it out would release energy. And don't worry, we're going to talk about that later. Okay, so we released a proton. Awesome. And now we're going here. We're sending this here. Let's highlight a couple terms because there are a lot of words here. So the thing that we're using to pump the proton into proton pump is the cytochrome B6/F complex, which that's just what this is called. If you want, you can call this B6/F, but you're going to want to remember the cytochrome thing. But just like to label it, like what is this thing? It's that. It looks like a stretched out football. And then this thing is our photosystem 2. This is our photosystem 1. And it has pigment molecules, like chlorophyll, and it has 2 chlorophyll A molecules specifically. That's what's taking our electrons and absorbing our energy from the pigment molecules. And we have our antenna complex, which is just like this whole region. And then our reaction center, which is the point where we are taking this, breaking this water and pushing it through the 2 chlorophyll A molecules and having it absorbed by our electron acceptor. Okay, cool. Oh, and this is PQ plastoquina. Okay, moving on. So once it gets to plastocyanin, PC, our bestie over here, which I'm gonna draw an arrow all the way over here. Plastocyanin. Another thing, and it's like, yep, that's the thing. We're just gonna move on. You don't really have to know. Okay, it's gonna do a very similar thing as before. Almost exactly the same. So there's 2 chlorophyll A molecules here. I'm not going to label it this time. You're just going to have to remember that. And if you don't, then that might be a problem because they're literally the same. So what's happening here is we have some light energy, photons coming in, and it's bouncing around, absorbing, going from one molecule to the next, and that'll be absorbed by the 2 chlorophyll A molecules, and then sent up with our 2 electrons. So this whole like arrow thing situation that's like following by, this represents the electrons that we got from the water. This arrow is the energy transmitted from the photons of the sun. So when we see that happening here, but this long like path that we're following the whole time is the electrons that we're exciting or using the energy from the water. So here we excite it, and then we get to use the high energy to pump a proton in, and then it's low energy again, and then we excite it again with all the light energy from photosystem 1, and then you'd never guess, it gets accepted by the electron acceptor. Wow. Oh yeah, I didn't highlight her. Nope. I didn't highlight her. She's the electron acceptor, and I'm not writing it again because she is still the electron acceptor over here. And now she gets to go to this guy, and you say, what's this guy? This is FD, my boy FD. And you're like, what is that? Why do we have so many 2-lettered things? This is ferredoxin. They all have names that you're like, I don't know what that is, and you don't have to. Yep, okay, moving on. And what happens here is we're turning NADP plus and a proton. So using another proton, and we're turning that into NADPH. And you're like, okay, I don't know what's going on. Why are we doing this? Don't worry about it. You will see why we need all this NADPH later. So we are not making glucose yet. We are making the stuff we need to make glucose later. And one of those things is NADPH. So that's actually really important. Oh. So the two things that we've done so far is we've put more protons in here and we've made it NADPH. And that's really essential for the stuff we'll need to make glucose later. So the next thing that we need is for this guy, who we call ATP synthase, to synthesize some ATP, which, nice name. All these guys have nice names. Pretend you can read this. This is synthase, synthase. Okay, so we're not gonna follow the arrow going to ATP synthase, because what just happened is we have NADP plus and a hydrogen ion, so a proton, and we're making NADPH. So basically we're adding an electron here, and then there's no plus sign. So there's 2 plus signs here, no plus signs here, and that's because we added 2 negative signs in the form of 2 electrons. So we got 2 electrons that we're following all the way through here. We pump in a proton, we keep going, and then boom, we lose it. It is put into the NADP plus and H plus to make NADPH by ferredoxin. Okay, so ATP synthase doesn't have the electrons. ATP synthase does something else. What does she do? So there's all these protons in here, right? Let's draw another one. And what ATP synthase is going to do is it's going to do what we were talking about before, where we let the proton out. Oh, where are we going to draw this? I'll just put it right here. So yeah, these protons really want to escape. And so what ATP synthase is going to do is it will let a proton out, and that's going to release a ton of energy. And there's more details in how ATP synthase kind of changes shape, but for now, all we're going to talk about is what's going to happen is we're going to use that energy it releases, which is it turns ADP and an inorganic phosphate into ATP. ATP is awesome. ATP is our unit of cellular energy in the cell, in life. You need this. This is like money. This is where you get your energy. This is how you do stuff. So ATP, super valuable. So hooray, we have ATP. So ATP and NADPH are the products of the light reactions of photosynthesis. So that's what this is. We need light that are really important to make the glucose later. So everything that we just talked about is the light reactions. So we can say light reactions. So, or the light dependent reactions if you want to write that. You can. Dependent. And obviously that's because it depends on light. If you don't have sun, then you can't boost these electrons to a higher energy level and you can't have all this happening. So this whole sequence is light dependent. It relies on the light. You cannot have it without the light. And in our next little episode, we'll talk about the Calvin cycle. So what you really want to remember from this is the products of the light reactions that we'll need for later is we make NADPH and ATP. Yay! We will need that. NADPH, ATP. And it looks like I forgot to highlight plasticinin, and I'll highlight H2O because that's important. You will almost certainly have to remember that photosystem 2 is the one that splits H2O. You know, like that's not happening over here. We have our electrons, you know? Our electrons are traveling all the way through here until we put them into this thing to make NADPH. So we don't need to split that again. And this will emit, remember, as gas and exit through the stomata. So a little review. Photosynthesis, it's only done in autotrophs like plants, cyanobacteria, and protists, also known as algae. And the whole goal here is to make glucose. So we'll take light like we did just now, and water, like we just did. We took the water, and CO2, which that's later. Spoiler alert, you don't know this yet, but you will, and it's very important. And then we make our glucose, and we have some water emitted as gas and some oxygen is emitted as gas. So that's here. We're emitting some oxygen as gas, and you don't have to worry about like, oh, well, aren't these protons being emitted? That's just adding to the lovely lumen that's very acidic, and that's increasing our proton gradient, which makes ATP synthesis job a lot easier because then it can let out a hydrogen and make some ATP. Okay, so hopefully you enjoyed this lovely little lecture on the light reaction to photosynthesis and a little introduction to, what's a chloroplast. And in our next episode, we will learn about the light independent reactions, the Calvin cycle, and it's gonna be amazing. Just you wait. All right, bye guys.